Understanding cells, the basic units of life. Cells are often referred to as the fundamental units of life, and for good reason. Every organism, from the simplest bacteria to complex multicellular creatures like humans, is composed of cells. Understanding what makes cells so essential to life is a key concept in biology and is fundamental for any university student pursuing a degree in the biological sciences. What makes cells the basic units of life? Cells are considered the basic units of life because they possess all the necessary components to carry out the functions essential for survival and reproduction. They are capable of metabolism, which involves the transformation of energy and materials to sustain the organism. Importantly, cells can reproduce independently, allowing for growth and healing in multicellular organisms. This autonomy underscores their status as the building blocks of life. Without cells, organisms cannot exist. Each cell can maintain homeostasis, allowing it to regulate its internal environment, respond to external stimuli, and preserve a stable state. This adaptability is vital for survival, as it enables organisms to thrive in various environments. Additionally, cells are involved in the organization of biological structures. They collaborate and communicate through signaling pathways, forming tissues and organs in multicellular organisms, and allow for the complexity and specialization we see in higher forms of life. Exploring cell structure, prokaryotic versus eukaryotic cells. When examining cells, they are generally categorized into two broad types, prokaryotic and eukaryotic. Prokaryotic cells are the simpler of the two and lack a nucleus. Their genetic material is not enclosed within a membrane, instead, it is found in a region called the nucleus. Prokaryotic cells are typically smaller than eukaryotic cells and include organisms such as bacteria and archaea. Despite their simplicity, they possess a remarkable ability to adapt to various environments, including extreme conditions. Key features of prokaryotic cells include a cell membrane, which provides protection and helps regulate what enters and exits the cell, a cell wall, offering structural support, and ribosomes, essential for protein synthesis. Some prokaryotic cells also have flagella or pili that assist in movement and adherence to surfaces, further showcasing their diverse functions. Eukaryotic cells, in contrast, are more complex. They contain a defined nucleus where the genetic material is housed, and they consist of various membrane-bound organelles such as mitochondria, the endoplasmic reticulum, and the Golgi apparatus. These organelles allow for compartmentalization of cellular functions, making eukaryotic cells more efficient in carrying out metabolic processes. Eukaryotic cells can be single-celled, like yeast and protists, or multicellular, such as plants, animals, and fungi. Enzymes, accelerators of biological reactions. A crucial aspect of cellular function is how cells interact with their environment and conduct numerous biochemical reactions. Enzymes play a significant role in this process by acting as catalysts that accelerate chemical reactions within cells. They lower the activation energy required for reactions, allowing processes to occur more quickly and efficiently. Each enzyme is specific to a particular reaction or type of substrate, ensuring that metabolic pathways function properly and efficiently. For instance, digestive enzymes help break down food into smaller molecules that the cell can use for energy and growth. Without enzymes, biochemical reactions would occur too slowly to sustain life, highlighting their vital importance to cellular processes. As you dive deeper into the complexities of biology, understanding cells, both their structure and their functions, will provide you with a comprehensive foundation for exploring the vast array of life forms on our planet. Embrace this knowledge as it opens the door to advanced biological concepts and interconnections that define life itself. Structures and functions of prokaryotic and eukaryotic cells. Understanding the structures and functions of prokaryotic and eukaryotic cells is essential for grasping the fundamental principles of biology. Let's dive deeper into the details of these two cell types. Prokaryotic cells. Prokaryotic cells are the simplest and most ancient forms of life on Earth. They lack a defined nucleus and other membrane-bound organelles, which distinguishes them from eukaryotic cells. Here's a closer look at the structures found in prokaryotic cells and their respective functions. Cell wall. The cell wall is a rigid structure that surrounds the cell membrane, providing shape and protection against mechanical stress and osmotic pressure. In bacteria, the cell wall is made primarily of peptidoglycan, a polymer consisting of sugars and amino acids. Gram-positive bacteria have thick peptidoglycan layers that retain crystal violet dye during staining, while gram-negative bacteria have a thinner layer surrounded by an outer membrane. Cell membrane. The cell membrane is a phospholipid bilayer that controls the movement of substances in and out of the cell. It is semi-permeable, allowing selective transport of ions and molecules through protein channels and carriers. This membrane also hosts various enzymes and transport proteins, facilitating biochemical processes. Cytoplasm. Cytoplasm is the gel-like fluid inside the cell, where most cellular processes occur. It contains water, salts, and organic molecules, serving as a medium that supports the organelles and structures within the cell. Nucleus. Unlike eukaryotic cells, prokaryotic cells do not have a membrane-bound nucleus. Instead, their genetic material, usually a single, circular strand of DNA, is located in the nucleus region. This DNA carries the instructions for cellular function and reproduction. Ribosomes. Ribosomes are the sites of protein synthesis in all cells. In prokaryotes, ribosomes are smaller than those found in eukaryotic cells, 70s in size compared to 80s in eukaryotes. They translate mRNA into polypeptides, enabling the production of proteins necessary for cell function. Flagella and pili. Flagella are long, whip-like structures that enable prokaryotic cells to move. They are composed of the protein flagellin and rotate like a motor to propel the cell. Pili, on the other hand, are short hair-like appendages that allow for attachment to surfaces, facilitate bacterial conjugation, transfer of DNA between bacteria, and play roles in biofilm formation. Plasmids. Plasmids are small, circular DNA molecules that exist independently of the chromosomal DNA. They can carry genes that confer advantageous traits, such as antibiotic resistance. Plasmids can be transferred between bacteria, contributing to genetic diversity. Eukaryotic cells. Eukaryotic cells are more complex than prokaryotic cells and have a greater variety of organelles, which allows for compartmentalization of cellular functions. Eukaryotic cells can be divided into animal cells and plant cells, with some structures being unique to each. Nucleus. The nucleus is the defining feature of eukaryotic cells and is surrounded by a double membrane called the nuclear envelope. It houses the cell's genetic material, DNA, organized into linear chromosomes. The nucleus also contains nucleoli, where ribosomal RNA synthesis occurs, and plays a vital role in regulating gene expression and cell division. Endoplasmic reticulum, The endoplasmic reticulum is divided into two regions, rougher and smoother. Rougher, studded with ribosomes, Ruffer is involved in the synthesis of proteins destined for secretion, incorporation into the plasma membrane, or for use in lysosomes. The ribosomes translate mRNA into polypeptide chains, which enter the ruffer for folding and modification. Smoother, lacking ribosomes, smoother is involved in lipid synthesis, metabolism of carbohydrates, and detoxification of drugs and poisons. It also plays a role in the storage of calcium ions, particularly in muscle cells. Golgi apparatus. The Golgi apparatus is a stack of flattened membrane-bound sacs known as cystinae. It modifies, sorts, and packages proteins and lipids synthesized in the rough and smoother for secretion or delivery to other organelles. The Golgi is crucial for glycosylation, the addition of sugar moieties to proteins and lipids, which is essential for their function. Mitochondria. Mitochondria are known as the powerhouses of the cell. They are double-membraned organelles that produce adenosine triphosphate, ATP, the energy currency of the cell, through oxidative phosphorylation. Mitochondria contain their own circular DNA and ribosomes, allowing them to produce some of their own proteins, supporting the endosymbiotic theory. Chloroplasts, in plant cells. Chloroplasts are double-membraned organelles specific to plant cells and some protists. They contain chlorophyll, the pigment responsible for capturing sunlight for photosynthesis, converting light energy into chemical energy stored as glucose. Like mitochondria, chloroplasts have their own DNA and replicate independently. Lysosomes. Lysosomes are membrane-bound organelles containing hydrolytic enzymes necessary for intracellular digestion. They break down waste materials, old organelles, and foreign pathogens through a process called autophagy. This function is vital for cellular recycling and waste management. Peroxisms. Peroxisms are small, membrane-bound organelles that contain enzymes for the oxidation of fatty acids and the detoxification of harmful compounds, such as hydrogen peroxide, which is produced during certain metabolic reactions. They play a crucial role in lipid metabolism and maintaining cellular oxidative balance. Cytoskeleton. The cytoskeleton is a network of protein filaments and tubules that provides structural support, shape, and the ability to move. It is composed of three main components: microfilaments, actin filaments, intermediate filaments, and microtubules. The cytoskeleton is also involved in intracellular transport and cell division. Plasma membrane. Similar to prokaryotic cells, eukaryotic cells have a plasma membrane composed of a phospholipid bilayer that regulates the movement of substances. However, eukaryotic membranes contain various proteins for signaling and transport, and they can have cholesterol, which helps maintain membrane fluidity. Prokaryotic cells, simplicity and adaptability. Cell wall structure. The composition of the prokaryotic cell wall is crucial for survival. In many bacteria, the peptidoglycan layer provides mechanical strength and protection from osmotic pressure. The molecular structure of the cell wall can influence antibiotic susceptibility. For instance, gram-negative bacteria have an outer membrane that contains lipopolysaccharides, which can act as an endotoxin and is more resistant to certain antibiotics as compared to the thick peptidoglycan layer of gram-positive bacteria. Reproduction. Prokaryotic cells reproduce asexually through binary fission, a straightforward process where a single cell divides into two identical daughter cells. This rapid reproduction allows for quick population growth and adaptation to environmental changes. Genetic recombination can occur through processes such as transformation, uptake of foreign DNA, transduction, transfer by viral infection, and conjugation, direct transfer via pili. Metabolic diversity. Prokaryotes exhibit extraordinary metabolic versatility. They occupy various ecological niches, from extreme environments, such as hydrothermal vents and salty lakes, to more typical habitats. Some bacteria are photosynthetic, like cyanobacteria, while others are chemosynthetic, deriving energy from chemical reactions involving inorganic substances. This metabolic diversity allows them to exploit resources that may not be accessible to eukaryotic organisms. Endospores. Certain bacteria can form endospores in response to harsh environmental conditions. Endospores are highly resistant structures that ensure the survival of the bacterial genome during drought, heat, or nutrient depletion. When conditions become favorable again, the endospore can germinate and produce a new functional bacterium. Eukaryotic cells, complexity and specialization. Eukaryotic cells are distinguished by their complexity and compartmentalization, allowing them to perform a variety of specialized functions. Next slide will include detailed features of eukaryotic cells, focusing on their structures and functions. Nucleus and genetic regulation. The nucleus serves as a control center for cellular activities. The nuclear envelope is composed of two lipid bilayers, controlling the exchange of materials between the nucleus and cytoplasm through nuclear pores. Chromatin, a complex of DNA and histone proteins, condenses to form chromosomes during cell division, ensuring the proper distribution of genetic information to daughter cells. Additionally, the regulation of gene expression in the nucleus plays a critical role in cellular differentiation and response to environmental changes. Membrane-bound organelles. The presence of membrane-bound organelles allows for compartmentalization of different metabolic processes, increasing efficiency. Mitochondria, known as the powerhouses of the cell. Mitochondria are critical for ATP production via the Krebs cycle and oxidative phosphorylation. The inner membrane is highly folded into crista, expanding the surface area for chemical reactions. Mitochondria also have a unique role in apoptosis, programmed cell death, releasing cytochrome C that initiates the apoptotic pathway. Chloroplasts. In plants and some protists, chloroplasts are involved in photosynthesis, converting light energy into chemical energy stored in glucose. They contain thylakoids, where light-dependent reactions occur, and stroma, where the Calvin cycle takes place. Like mitochondria, chloroplasts contain their own DNA and ribosomes, highlighting their endosymbiotic origins. Intracellular transport. The endoplasmic reticulum, both rough and smooth, plays a key role in processing and transporting proteins and lipids. For example, proteins destined for secretion are synthesized in the rougher, glycosylated, and then transported to the Golgi apparatus for further modifications and sorting. The smoother, rich in enzymes, aids in lipid metabolism and detoxification, indicating the specialization of eukaryotic cells in maintaining homeostasis. Cytoskeleton dynamics. The cytoskeleton provides structural support and enables intracellular transport via motor proteins that travel along microtubules. Additionally, the cytoskeleton is essential for cell division. During mitosis, microtubules form the mitotic spindle that separates chromosomes to opposing poles of the cell. Cell communication and signaling. Eukaryotic cells utilize a range of signaling mechanisms to communicate and respond to their environment. This includes receptor-mediated signaling through membrane proteins. Examples include hormone signaling, neurotransmitter reception, and response to environmental stress signals. The complexity of signaling pathways allows eukaryotic cells to integrate diverse signals and coordinate responses, which is critical in multicellular organisms. Cellular specialization. In multicellular organisms, eukaryotic cells can differentiate into various cell types, each specialized for specific functions. For instance, red blood cells are designed for oxygen transport, while neurons have features that support rapid signal transmission. This specialization contributes to the overall functionality of tissues and organs, enabling complex organisms to perform higher-order biological functions.